So we'll take some questions at the end for all of our speakers. And I'd like to now invite Peter from the German Cancer Research Center, and I will let you introduce yourself also.
Thank you very much. So I think I wanted to present some thoughts on data sharing through the experience that I have in particular as an active member in the ICGC, the International Cancer Genome Consortium. And can I? Yeah. Great, thanks.
There we go.
Okay. And you know, this consortium had started in 2008, basically, and at the moment we have 90 projects in this. The first phase of this project comes to an end. And the databases are actually being filled now. We are at the goal of more than 20,000 that is being reached. Lincolnstein had made a summary of this. But it is not so easy as one thinks. If we talk about uploading of scientific data, I think the EGA could probably give the same experience. It still is dominated by the right of first-time publishing, first-time intellectual property rights, and so on and so on. Things are delayed. If I've heard this fantastic talks this morning about what we can do with the data, the access to the data is very well being taken care of, and all of these things. We need full databases to really apply all of these things. You know, that was based on the idea not just to do genomics, but also to do transcriptomic and clinical data in particular. And obviously data sharing is a major part of this. Without this, it doesn't work. Now, more recently, in recent years, as you know, a group within ICGC started to say we should look across entities, different tumor types, and get close to 3,000. At the end, it was 2,800 and some cancer genomes, and learn across the genomes many things of tumor biology, possibly also clinically. And this was one of the largest genomic experiments in terms of data pursued today still. And the steering committee from which I received some of these slides is listed here. And the idea obviously was we should also look into non-coding part of the genome, into pathogens, into mutation signatures, and so on. I don't need to go into the details. Many of you are active in this consortium and know about it. And it was possible at the end to come up with 2,834 cancer genomes, and the important part why I'm showing this, they come from 14 different jurisdictions. And you see the distribution by the different projects from the different countries. But that, of course, produced a number of issues, and one issue, certainly, it was underestimated how much capacity one needs. At the end, it was a mixture of cloud computing and academic centers that did this. Again, I should not go too much into the details here, but certainly there was a lot of discussion going on. If it is clouds, and that seems to be safer, how about public versus private clouds? How about academic versus commercial clouds? And certainly different countries had different perspectives and different results of that. Within Germany, where I'm from, we had some legal advice that also considered, for instance, what is the real world of jurisdiction? Do we have dozens of lawyers that would eventually go against one of these big cloud providers? And this is in the real world a concern. And of course, a concern was who are the public and commercial cloud providers, and can one trust all of them? That is still an ongoing discussion. Nevertheless, I think it was great that this came to a very big success, and you will probably later this year have quite a big wave of papers that come from Peacock. And this, as you can see, we have a mixture of academic and non-academic clouds, including commercial clouds, that are allowed to continue and have this a very successful overall network. And actually part of this is planned to become interoperable with the European Open Science Cloud, which would be a topic in itself. Many of you in the audience know much more about it than I do. To switch briefly, I was responsible for pediatric brain tumors within ICGC, and we, as a spin-off of all of these activities, we did quite a number of translational trials in Heidelberg and somewhere else. And the workflow is such that we can do a full personalized oncology trial with the initial time from taking samples to the molecular tumor board that discusses novel treatment options for an individual patient within 3 weeks. And examples are sometimes fascinating. This is a child that had an inoperable large tumor. It was a fusion gene detected where we have an inhibitor against the kinase. And this could be taken away after the successful diminishing during this treatment by the surgeon. As very often, tumor came back, but even the new tumor is sensitive to ALK inhibitors. That's the kind of success. And similar to what other groups and other trials show within this INFORM trial of relapsed patients in pediatric oncology, we have this 3-week achievement, and we have about 60% of novel actionable targets based on the whole procedure. And if you think about clinical response, we talk about, depending on the tumor type, between 25 and 40% in those cases where off-label use or compassionate use was performed. However, a common feature is, as you can see here, this is another child. We found a particular fusion again that we had found at the same time, and then the tumor after surgery could not be fully removed. It was fully removed, and the clinical situation became very good with such an inhibitor, but after not too long the patient died. And what we do have is this phenomenon of resistance. Whether this was during treatment or a clone that was just too small at the beginning cannot be shown at this particular case, but it tells us we have to look at tumor heterogeneity, we have to do longitudinal studies, and this has a lot to do with the problem, with some aspects of data sharing, which I'm mentioning at the end again. This is a study on breast cancer, and as you can see, this is high-risk breast cancer, mostly metastasized. And after going through the whole procedure to the board, these patients are basically introduced into other treatment arms. If you look at the very first series of patients of last year, and then you see we have quite a high number of cases where we have germline variants. If you look at the BRCA1 or BRCA2, 8 of the 54 had them. 4 of them were known to be pathogenic. But this is actually the information that we don't know how to deal with, because these are unclassified variants and they usually do not have an impact on any decision. This is the information, as you can see it in the molecular tumor board. Everything that is red is a potential novel target. If you look here, we have germline mutations, and we don't know really in many of these cases how we can deal with them. So far, they are not being dealt with when they are not known to be of pathogenic relevance. Is that suitable that we do that? In addition, there are treatment trials where patients here could be enrolled where we attack so-called BRCA of a tumor, and these are treated with PARP inhibitors. And this could be a confirmatory finding for this decision to attack BRCA, a particular pattern in the tumor genome, but we cannot use this information for it. And what we really need is something that the next phase of ICGC, called ICGC Argo, for accelerated research in genomic oncology, plans to do. We want to have large databases. We want to have data for at least 100,000 cancer patients. And in contrast, even though it was initially announced we would do in the first phase a lot of clinical data, there was only in a very few studies sufficient amount of clinical data to look further into that. Here this will change. We will look into the cases where we have metadata and clinical information. And the questions that are being answered here are: how do we use current treatments better? How does a cancer change over time, as I just mentioned? And how do we practically implement this in the healthcare system? And eventually exploit it for other means like preventive efforts and so on. And if we talk about metadata, it's so little data that we really talk about. The working group that I'm heading within ICGC Argo had come up with this type of data. We talk about altogether just about some 30 data points. They are in each clinical trial, almost in each clinical trial they are documented. And this is now a must, and this will allow us to ask the questions which I tried to bring up with a few examples, and which I would like to summarize again in this slide. We are being asked, so what are the benefits, what is the added value, is that you now do this just more cases but with clinical data? And I think it is very important that a doctor who sits in Canada and has such a variant of BRCA2 at his patient, or another type of a variant, he would like to know whether a doctor in Australia, I should probably not take only English-speaking countries, Ewan, right? So let's say in Japan, has the same finding. He had also the possibility to prioritize it as a target. Is such a rare molecular variant actually such that we have already drugs available, and we had some treatment somewhere, and maybe even some clinical response somewhere? If we really want to translate these findings, we have, in a fast matter, we need to do this worldwide. There are too many rare events that we have. And I think it's quite important that we all combine our efforts for this. It is important, was this already tried at the same tumor type or in a different tumor type? We know that some BRAF inhibitors do not work, even though it's the same mutation that they attack in certain tissues. What are the clinical consequences if we do combination treatments, which we need if we want to attack this type of resistance that I try to present to you? How about the ethnic background? Does it play a role, for instance, in adverse drug effects that can be found in very many different tumor entities? And obviously the situation with the familiar background and predispositions that we have in here. So the sharing certainly has the issues on we look at personal data, as we all know very well in this room, and even if we try to restrict it as much as possible, for most of the reasons why we want to share the data, we need more than just the very few mutations. So it's not just the extracted information that we can deal with. We need comprehensive genomes, at least to a certain extent, and we have to deal with the fact that these data will allow re-identification of patients based only on those data. There's a lot of discussion about anonymization versus pseudonymization. As you know, anonymization could be valuable for many of the internationally accessible parts of such a database. And one could, I think, subdivide this into certain areas. And germline information, we already talked about this. And we also talked about what is coming up now and has come up with GDPR. It had been the topic here and will be a topic further during this meeting. There is a lot of conclusions from this GDPR. Many have to do with how the consent forms should look like. We talk a lot about a right of access to data, right of correcting data, right to erasure data, records of processing activities, but we do not talk so much about what I want to show you in the next slide, this right from benefiting actually from scientific progress. And this is what Partha in the film this morning said briefly. We should try to take advantage of such human right formulations. It is Article 15 in the United Nations Human Rights List. There are lots of elements being developed. You can read the reports of the rapporteurs. And on the other hand, if you look, this is very general again, if you look what they are linked to, They are linked from Article 15 in particular to Article 27. And this is basically the right to participate in cultural life. And if you go then through the main points here, you find phrases like access by everybody, opportunities for all to contribute, participation of individuals and communities in decision making, which is certainly right and possible as long as we define very well who is behind that and what will be the procedures for that. And therefore, I would like to close by saying we should take the advantage, as Partha said. We should look for a good balance. We should press for as much of exploiting this benefit from scientific progress and its applications. And we have to go further than, for instance, in particular in my country, the personal data protection is being handled at. Thorough discussions are needed. I think one rule that should go always behind these is we should be as open as possible to allow as much of cross-fertilization across the globe and be as only as closed as necessary. And with that, I would like to finish my thoughts. Thanks.